Hey guys! Happy Hanukkah to all the Jewish people out there, and happy Sunday to everyone else who's not celebrating Hanukkah. So this is part 3, and this is going to be the last video for chapter 12. So let's get started. So cytologists, scientists that study the structure and function of cells, and that includes chromosomes as well, have discovered many structural rearrangements in chromosomes, but the most common ones are inversions and translocations. Chromosome inversions are the detachment, 180-degree rotation, and then the reinsertion of part of a chromosome. They usually occur through mutated DNA sequences that have the ability to direct this process with the help of enzymes. Since inversions only change the orientation of genes, they have milder effects than the loss or gain of genes through an haploidy. But if they do affect a specific gene sequence, many negative effects can result. There are 2 different types of inversions, pericentric and paracentric. Pericentric is where an inversion includes the centromere. So let's stop right there, and I'm going to show you pericentric inversion. So I'm going to step aside, and as you can see, we have a chromosome. The centromere is right here. So we have the segment of the chromosome, and I just showed you the segment. So here's the centromere, and this is cut. So imagine it being cut because it's being removed, and then it's being removed, and then it's rotated. So it goes from B to C to CB, and then it's reinserted as ACBD instead of ABCD. So that's pericentric, and so remember it includes the centromere. So see, the centromere comes with it, and then it gets flipped, so the centromere stays. So wait 1 second, and we'll talk about pericentric.
So as I just mentioned, there are 2 types of inversions, pericentric and pericentric and paracentric. They're so similar, I don't know why they decided to name them such similar names. Pericentric is where an inversion includes the centromere, and then paracentric is outside the centromere. So as you can see, you have the chromosome, here I'm going to point, and then you have the little sequence. This is the cut, is cut, disattached, I guess. Is that a word? I think I used it before. Yeah. It's removed, rotated, so you go DE, now it's ED. And so then it's reinserted as ABCED versus ABCDE. So that's it.
Translocations are what happens when a segment of a chromosome is detached and reattached to a non-homologous chromosome. They usually have devastating effects, like certain cancers and schizophrenia, but genetic information is not normally lost. So a translocation where no genetic material is lost is known as a reciprocal translocation. So let me show you the board behind me. So as you can see, I have a chromosome, and it's a chromosome. It's not with its So chromosome, another chromosome, non-homologous, so they're not a pair, completely unrelated, okay? See this little box? That's just representing where the changes are going to be. So this piece of the chromosome is detached and then placed here, and then this piece of the chromosome is detached and then placed there. Makes sense? And also no genetic information is lost.